The farthest distance a human body could physically travel from Earth before its biology irreversibly fails is approximately 4.3 light-years. That distance corresponds to the nearest star system beyond our Sun, Proxima Centauri. But the limit isn't set by the distance itself. It's set by the unavoidable interaction between human cells and the radiation environment of interstellar space. In 2013, NASA's Voyager 1 spacecraft became the first human-made object to cross into interstellar space, measuring radiation levels of approximately 0.3 to 0.4 nanosieverts per second from galactic cosmic rays alone. For context, that exposure rate, sustained over a multi-decade journey, would accumulate a dose exceeding 300 millisieverts, which is roughly 150 times the average annual background radiation dose on Earth. At doses above 100 millisieverts, the probability of radiation-induced cancer increases measurably. Above 1000 millisieverts, acute cellular damage begins, and above 4000 to 5000 millisieverts, delivered over a short period, survival becomes unlikely even with medical intervention. These are not theoretical projections, they are experimentally verified thresholds derived from observations of radiation exposure in nuclear accident survivors, medical patients, and astronauts monitored during spaceflight. If you enjoy this kind of evidence-based content, like the video, subscribe to the channel, and turn on notifications. There are many other videos on the channel built the same way, and if this topic interests you, you'll likely enjoy what's already there. The question of how far a human could travel is not primarily a question of propulsion, fuel, or time. It is a question of cumulative exposure. The human body evolved under a specific set of conditions: atmospheric shielding that blocks most high-energy particles, a magnetic field that deflects charged radiation, and a radiation environment that delivers roughly 2 to 3 millisieverts per year at sea level. Once you leave the magnetosphere, those protections vanish. The only barrier between your cells and the cosmic radiation flux is whatever material surrounds you. And material shielding, while effective against some particles, cannot fully stop the highest energy galactic cosmic rays. Galactic cosmic rays are atomic nuclei, primarily protons, that have been accelerated to near light speed by supernova remnants, pulsars, and other high-energy astrophysical processes. Their energies range from millions to billions of electron volts. When one of these particles strikes a spacecraft hull, it doesn't simply stop. It fragments, producing secondary particles, neutrons, pions, gamma rays, that scatter in all directions. Some of these secondaries are more biologically damaging than the original particle. This phenomenon, known as secondary radiation, was measured directly aboard the International Space Station using dosimeters placed at various locations. The data, published in multiple studies between 2000 and 2020, show that shielding beyond a certain thickness can actually increase the radiation dose inside a spacecraft, not decrease it. The dose rate inside the ISS averages approximately 150 to 200 microsieverts per day, depending on solar activity and orbital altitude. That's roughly 50 to 80 times higher than the surface of Earth. Astronauts on 6-month missions accumulate doses between 50 and 150 millisieverts. These values are well documented through personal dosimeters worn continuously, and cross-verified with tissue-equivalent phantoms equipped with radiation sensors. Long-term studies of astronauts who flew during the Apollo, Shuttle, and ISS eras indicate elevated rates of cataracts and possibly cardiovascular disease, though sample sizes remain small. What is clear from the data is that the longer the exposure, the greater the cumulative risk. In interstellar space, far from the Sun's heliosphere, the radiation environment is worse. The heliosphere is a bubble of charged particles emitted by the Sun that extends roughly 120 astronomical units in the direction of the Sun's motion through the galaxy. This bubble provides some deflection of lower-energy galactic cosmic rays. Inside it, radiation is modulated by the solar cycle. During solar maximum, increased solar wind pressure pushes cosmic rays outward, reducing the flux. During solar minimum, the opposite occurs. Voyager 1 measured this transition directly. When it crossed the heliopause in August 2012, its instruments detected a sudden increase in high-energy particles and a corresponding drop in solar particles. The galactic cosmic ray flux increased by a factor of 2 to 3 compared to measurements taken just inside the heliosphere. If a crewed spacecraft were to follow a similar trajectory, the crew would experience this increase firsthand. And unlike Voyager, which is a machine insensitive to ionizing radiation, a human crew would face continuous cellular damage. Ionizing radiation damages DNA in two primary ways. The first is direct ionization, where a particle strikes a DNA molecule and breaks a chemical bond. The second is indirect damage, where the particle ionizes a water molecule, creating reactive oxygen species, free radicals, that then attack nearby DNA. Both mechanisms have been studied extensively in laboratory settings using particle accelerators that simulate cosmic rays. Experiments using beams of iron nuclei, which represent a small but highly damaging fraction of galactic cosmic rays, showed that a single traversal through a cell nucleus can cause clustered DNA breaks that are difficult for cellular repair mechanisms to fix accurately. The human body contains trillions of cells, each with repair mechanisms refined over billions of years of evolution, but those mechanisms evolved in an environment where ionizing radiation was rare. A typical cell on Earth experiences a DNA-damaging event from background radiation perhaps once every few weeks. In space, that rate increases to several times per hour. Most damage is repaired correctly. Some is not. Misrepaired breaks can lead to mutations. If those mutations occur in genes that regulate cell division, cancer can result. If they occur in germ cells, they can be passed to offspring. If they occur in critical tissues like bone marrow or the lining of the digestive tract, they can impair the production of blood cells or cause chronic inflammation. Astronauts aboard the ISS are protected, to some degree, by the residual influence of Earth's magnetosphere, which deflects some charged particles even at orbital altitudes. They are also exposed for limited durations. A crewed mission to interstellar space would have either protection. The journey to Proxima Centauri, even aboard a spacecraft moving at 10% the speed of light, would take more than 40 years. At current propulsion capabilities, using chemical or ion drives, the journey would take tens of thousands of years. Both scenarios are biologically untenable without shielding or biological intervention. Let's examine shielding first. The most effective materials for blocking radiation are those with high hydrogen content, because hydrogen nuclei are single protons with no neutrons, minimizing secondary particle production. Water, polyethylene, and liquid hydrogen have all been studied as potential shielding materials. A study published in 2016 by researchers at the NASA Langley Research Center calculated that a water shield approximately 50 centimeters thick would reduce the galactic cosmic ray dose by roughly half. To reduce it to levels comparable to Earth's surface would require several meters of water equivalent, resulting in a massive increase in spacecraft mass. For a crewed interstellar mission, carrying that much shielding is impractical under known propulsion methods. Active shielding, using magnetic or electrostatic fields to deflect charged particles, has been proposed. The concept mimics Earth's magnetosphere. A spacecraft would generate a magnetic field strong enough to bend the trajectories of incoming protons and heavier ions, diverting them around the crew compartment. Laboratory experiments and computational models suggest this is theoretically possible. However, the energy requirements are enormous. Maintaining a field strength comparable to Earth's magnetosphere, across a volume large enough to protect a spacecraft, would require continuous power output in the megawatt range. No current spacecraft power system, whether solar panels, radioisotope thermoelectric generators, or even conceptual nuclear reactors, could sustain that output over decades. Furthermore, magnetic shielding is ineffective against neutral particles, such as neutrons, which are a significant component of secondary radiation. Another approach is pharmacological or biological intervention. Certain compounds, known as radioprotectors, can reduce cellular damage from radiation by scavenging free radicals before they attack DNA. Amifostine is one such compound, used clinically to protect healthy tissue during radiation therapy for cancer. Studies in animal models show it can reduce radiation-induced DNA damage by 20 to 40% when administered before exposure. However, it has significant side effects, including nausea, low blood pressure, and toxicity with prolonged use. Administering it continuously over years or decades is not feasible. Other compounds, such as antioxidants like vitamin E or selenium, have shown modest protective effects in lab studies, but their efficacy in humans exposed to chronic low-dose radiation remains unproven. Genetic modification has also been considered. Some organisms, such as the bacterium Deinococcus radiodurans, can survive radiation doses thousands of times higher than what would kill a human. This bacterium has multiple copies of its genome and highly efficient DNA repair enzymes. Researchers have identified specific genes responsible for this resilience. In principle, introducing similar mechanisms into human cells could enhance radiation resistance. Experiments with human cell lines in culture have demonstrated that overexpressing certain DNA repair genes can improve survival after irradiation. But translating this to whole organisms, especially humans, involves enormous technical and ethical complexities. Germline editing, which would be necessary to confer resistance to future generations, is currently prohibited in most jurisdictions and raises profound questions about unintended consequences. Even if shielding and biological interventions were perfected, there are limits imposed by other aspects of human biology. One is the central nervous system. Neurons, unlike most other cells, do not divide after maturity. This makes them resistant to some forms of radiation damage, because they don't undergo the cell division process where most radiation-induced errors become fatal. However, high-energy particles can still cause direct damage to neuronal structures. Experiments on rodents exposed to simulated cosmic rays showed deficits in memory, learning, and spatial navigation, even at doses below those that cause cancer. These effects appear to result from damage to synapses and the death of specific neuron populations in the hippocampus. A 2016 study published in the journal Scientific Reports exposed mice to beams of oxygen and titanium nuclei at energies typical of galactic cosmic rays. 6 months later, the mice showed impaired performance on memory tasks and increased anxiety-like behaviors. Post-mortem analysis revealed inflammation and reduced connectivity in brain regions critical for cognition. If similar effects occur in humans, and there is no reason to assume they would not, then even a well-shielded crew on a decades-long interstellar journey might suffer progressive cognitive decline. The social and operational consequences of this are difficult to quantify but potentially catastrophic. A crew unable to make complex decisions, operate systems, or respond to emergencies would be unlikely to complete the mission. Another biological limit is bone density. In microgravity, bones lose mass at a rate of approximately 1 to 2% per month. This has been measured extensively in astronauts using dual-energy X-ray absorptiometry before and after spaceflight. The loss is due to reduced mechanical loading. Bones constantly remodel themselves in response to stress. Without gravity, the signals that promote bone formation diminish, while bone resorption continues. Long-duration astronauts on the ISS undergo rigorous exercise regimens, using resistance devices to mimic the mechanical loading of gravity. Despite this, they still lose bone mass, particularly in the hips and spine. After 6 months, typical losses are 5 to 10%. After a year, losses can approach 15%. Some of this is recovered after return to Earth, but not all. In a multi-decade journey, cumulative bone loss could lead to fractures, impaired mobility, and chronic pain, even with exercise countermeasures. Artificial gravity, generated by rotating a spacecraft, would solve this problem. However, the engineering challenges are substantial. To produce a centripetal acceleration equivalent to Earth's gravity, a spacecraft must either rotate rapidly or have a large radius. Rapid rotation causes problems with orientation and motion sickness, due to Coriolis forces that affect fluid in the inner ear. A radius of several dozen meters, rotating at a few revolutions per minute, would provide 1 gravity of acceleration at the rim, while minimizing Coriolis effects. The ISS, for comparison, has a maximum dimension of about 109 meters, but it does not rotate. Building a rotating structure large enough for artificial gravity, strong enough to withstand the stresses, and capable of sustaining a crew for decades, is beyond current engineering. Muscle atrophy follows a similar pattern to bone loss. Without gravity, muscles don't need to work as hard to maintain posture or move the body. They adapt by reducing mass and strength. Astronauts can lose 20% or more of their muscle mass during a 6-month mission, despite exercising 2 hours per day. The heart, a muscle itself, also weakens. It becomes less efficient at pumping blood, because in microgravity, the body's circulatory system doesn't have to fight gravity to return blood from the legs to the heart. Upon return to Earth, astronauts often experience orthostatic intolerance, dizziness or fainting when standing, due to the heart's reduced capacity and changes in blood pressure regulation. These effects would be compounded over decades. Then there is the immune system. Multiple studies have documented changes in immune function during spaceflight. White blood cell counts shift. The activity of natural killer cells, which destroy infected or cancerous cells, decreases. Latent viruses, such as herpes simplex, Epstein-Barr, and varicella zoster, can reactivate in astronauts due to immune suppression and stress. This has been confirmed through viral shedding detected in saliva and blood samples collected during and after missions. The mechanisms are not fully understood, but they likely involve a combination of microgravity, radiation, isolation, disrupted sleep, and psychological stress. A crew on an interstellar mission would face all of these factors, continuously, with no possibility of early return. Psychological factors are equally significant. Humans evolved in social groups, with access to natural environments, varied stimuli, and the ability to move freely across large areas. Confining a small group of people inside a metal vessel, surrounded by vacuum and lethal radiation, for decades, creates conditions unlike anything our species has experienced. Analogues exist, such as polar research stations, submarines, and space station missions, but none approach the scale or permanence of an interstellar voyage. Crews in these environments report stress, interpersonal conflict, sleep disturbances, and depression, even on missions lasting months. Isolation, monotony, and the inability to leave or receive help create psychological strain that accumulates over time. The most detailed study of long-duration isolation is the Mars 500 project, conducted by the European Space Agency and Russia's Institute for Biomedical Problems between 2007 and 2011. 6 volunteers were sealed inside a simulated spacecraft for 520 days, the approximate duration of a round trip to Mars. They experienced sleep disturbances, decreased physical activity, and interpersonal tension. One participant developed a pattern of sleeping during the day and staying awake at night, despite the absence of external cues. Another became increasingly withdrawn. While the crew completed the simulation, the psychological toll was evident. Extending this to decades, or even generations, introduces uncertainties that cannot be fully predicted. A generational ship, where multiple generations live and die en route, is sometimes proposed as a solution. Children would be born, raised, and educated aboard the spacecraft, eventually taking over from their parents. But this introduces additional biological challenges. Human reproduction in microgravity has never been studied beyond basic cellular and animal experiments. Mice have been bred in space, but mammalian development is a complex process influenced by gravity at multiple stages, from fertilization to embryonic growth to birth. Fluid distribution, cellular orientation, and mechanical forces all play roles. Whether a human fetus could develop normally in microgravity is unknown. Even if reproduction succeeds, raising children in a confined, artificial environment, with no access to Earth's biosphere, raises questions about psychological and social development that have no empirical basis. There is also the question of genetic diversity. A small founding population, perhaps a few dozen to a few hundred individuals, would have limited genetic variation. Over multiple generations, inbreeding would become unavoidable, increasing the risk of genetic disorders. Population genetics models suggest that a minimum effective population size of several thousand is necessary to maintain genetic health over many generations. This is far larger than any spacecraft could realistically carry. Genetic engineering or artificial gamete storage might mitigate this, but these solutions remain speculative. Let's return to the central question, the farthest point a human could reach before biology fails. The answer depends on how we define failure. If failure means death from acute radiation exposure, the limit is determined by cumulative dose. A dose of 5,000 millisieverts, delivered over days to weeks, is lethal to roughly half of exposed individuals without medical treatment. This is known as the LD50 dose. If a spacecraft travels through an interstellar radiation environment at 0.3 nanosieverts per second, it would take approximately 530 years to accumulate 5,000 millisieverts, assuming no shielding. With shielding that reduces the dose by half, the time doubles to roughly 1,060 years. Even at velocities of 10% the speed of light, reaching Proxima Centauri would take 43 years, during which the crew would accumulate a dose of approximately 400 millisieverts in an unshielded scenario, or 200 millisieverts with modest shielding. This is below the acute lethal threshold but well above the level where cancer risk becomes significant. If failure means the inability to complete the mission due to cancer, cognitive decline, bone fractures, or immune compromise, the limit is more restrictive. These chronic effects accumulate over time and become debilitating long before they are lethal. Based on animal studies and astronaut data, it is reasonable to estimate that without artificial gravity and advanced radiation shielding, a human crew could not sustain biological function for more than a few decades in interstellar space. That corresponds to a maximum distance of a few light years, assuming near relativistic velocities, or far less with current propulsion. Now consider the practical realities. The fastest spacecraft ever built, the Parker Solar Probe, reached a maximum speed of approximately 163 km per second relative to the Sun, or about 0.05% the speed of light. At that speed, reaching Proxima Centauri would take roughly 8,600 years. No human crew could survive that duration. Even if we imagine a spacecraft capable of sustained acceleration at 1 gravity, which would provide both propulsion and artificial gravity, the energy requirements are staggering. Accelerating a spacecraft with a mass comparable to the ISS, roughly 420,000 kilograms, at 1 gravity for years would require energy far exceeding the output of any current or near-future power source. Nuclear fusion, often cited as a candidate, remains unproven for propulsion, despite decades of research. The Breakthrough Starshot project, announced in 2016, proposes an alternative, using ground-based lasers to accelerate lightweight probes to 20% the speed of light. These probes would be tiny, perhaps a few grams, carrying miniaturized instruments. They could reach Proxima Centauri in roughly 20 years. But scaling this concept to carry humans is not feasible. The laser power required to accelerate even a small crewed spacecraft would be measured in terawatts, far beyond any existing or planned installation. And decelerating at the destination, without fuel, is an unsolved problem. Another concept is the nuclear pulse drive, also known as Project Orion, studied in the 1950s and 60s. The idea is to detonate nuclear bombs behind a spacecraft, using a massive shock plate to absorb the energy and convert it into thrust. Calculations suggest this could achieve velocities of a few percent the speed of light. Models indicated it was theoretically possible to build such a spacecraft with 1960s technology. However, the Partial Nuclear Test Ban Treaty of 1963 effectively ended research into nuclear propulsion in space. Even if political and environmental objections were overcome, the biological effects of repeated nuclear detonations, even at a distance, would need careful study. The radiation from each detonation, combined with the interstellar background, could exceed safe limits. There is also the matter of navigation. At high velocities, even small particles become hazardous. A grain of sand 1 milligram in mass, striking a spacecraft traveling at 10% the speed of light, would release energy equivalent to approximately 450 kilojoules, comparable to a hand grenade. Interstellar space is not empty. It contains approximately 1 hydrogen atom per cubic centimeter on average, along with dust grains and larger debris. At relativistic speeds, collisions with this material would erode shielding, damage systems, and potentially cause catastrophic failure. Detecting and avoiding such particles at high speed is extremely difficult. Light-speed communication delays mean that by the time a hazard is detected, the spacecraft may have already passed it. Then there is time dilation. At velocities approaching the speed of light, time aboard the spacecraft slows relative to the rest of the universe, according to Einstein's theory of special relativity. This has been verified experimentally using atomic clocks flown on aircraft and satellites. At 10% the speed of light, the effect is small, roughly a 0.5% difference. But at 90% the speed of light, time aboard the spacecraft would pass at less than half the rate of Earth time. A journey that takes 50 years on Earth might feel like 20 years to the crew. This would reduce the biological burden of the journey, but it introduces a different problem: temporal isolation. By the time the crew reaches their destination, decades or centuries may have passed on Earth. Everyone they knew would be dead. The society they left might have changed beyond recognition. Communication, limited by the speed of light, would involve years of delay. A message sent from Proxima Centauri would take 4.24 years to reach Earth, and the reply another 4.24 years to return. This is not a biological limit, but it is a profound psychological and existential one. We must also consider the destination. Proxima Centauri is a red dwarf star, smaller and cooler than the Sun. It has at least 2 known planets, one of which, Proxima b, orbits in the habitable zone where liquid water could exist. However, red dwarfs are known for violent stellar flares, sudden increases in brightness and radiation output that can be thousands of times more intense than solar flares. These flares emit high-energy ultraviolet and X-ray radiation, as well as charged particles. Proxima Centauri flares regularly, with major events observed every few months. A planet in the habitable zone of such a star would be subject to intense radiation, potentially stripping its atmosphere and sterilizing its surface. If a human crew reached Proxima b, they would face an environment far more hostile than interstellar space itself. Even Earth-like planets around Sun-like stars may not be habitable. Habitability depends on many factors: magnetic field, atmospheric composition, temperature, water availability, geological activity, and the absence of severe radiation. We have identified thousands of exoplanets, but characterizing their surfaces and atmospheres requires spectroscopic analysis that is difficult with current telescopes. The James Webb Space Telescope, launched in 2021, can detect certain atmospheric molecules in the largest exoplanets, but detailed surface mapping remains beyond our capabilities. Sending a crew to an exoplanet without certainty of habitability would be extraordinarily risky. There is one final biological limit worth considering: the cellular aging process. Human cells can only divide a finite number of times before they stop, a phenomenon known as the Hayflick limit. This is due to the shortening of telomeres, protective sequences at the ends of chromosomes, with each division. After roughly 50 to 70 divisions, telomeres become too short, and the cell enters senescence or dies. This limits the regenerative capacity of tissues. While some cells, like stem cells, have mechanisms to maintain telomeres, the overall aging process is influenced by this limit. A person born aboard a generational ship would age normally, but the cumulative cellular stress from radiation, microgravity, and artificial conditions might accelerate aging or reduce maximum lifespan. No data exists on this, because no human has spent a lifetime in space. Radiation-induced senescence is a documented phenomenon. Cells exposed to ionizing radiation can enter a state where they stop dividing but do not die. These senescent cells secrete inflammatory molecules that damage surrounding tissue, a process called the senescence-associated secretory phenotype. In laboratory studies, mice exposed to radiation show increased senescent cells in multiple organs, along with symptoms resembling premature aging, frailty, cognitive decline, and organ dysfunction. If this occurs in humans on long space flights, it would compound the effects of microgravity and isolation. Let's synthesize what we know. The farthest point a human could reach, under current biological understanding, is constrained by radiation, bone and muscle loss, immune suppression, cognitive decline, psychological strain, and the practical limits of life support and propulsion. If we assume a mission with the best available countermeasures, modest shielding, artificial gravity via rotation, psychological support, and careful crew selection, a journey of a few decades might be survivable. That corresponds to a distance of a few light years, if propulsion technology advances dramatically. Proxima Centauri, at 4.24 light-years, sits near or beyond this limit. In practice, the first interstellar missions would likely be robotic. Machines do not require life support, do not age, and can be repaired or replaced. They can operate for decades or centuries, as Voyager has demonstrated. Human missions, if they occur at all, would follow only after extensive robotic exploration, technological advancement, and possibly biological modification. We also must acknowledge the possibility that interstellar travel, for biological humans, may never be practical. The distances are vast, the hazards severe, and the resources required enormous. Our species evolved to thrive on the surface of one planet, under specific conditions. Leaving that environment for extended periods, let alone permanently, pushes biology to its limits. Those limits are real, measurable, and based on decades of research in space medicine, radiation biology, and physiology. The evidence from Voyager, from the ISS, from particle accelerator experiments, from astronaut medical studies, from animal models, and from theoretical physics all point to the same conclusion. A human body can travel far, but not infinitely far. The constraints are written into the structure of our cells, the sensitivity of our neurons, the density of our bones, and the fragility of our DNA. Technology can extend those limits, but it cannot erase them. The farthest point is not defined by a single barrier but by the accumulation of many, each one measured, documented, and grounded in experimental reality. If we were to draw a line in space, marking the maximum distance a human could reach before biology fails catastrophically, that line would fall somewhere between the edge of the heliosphere and the nearest stars. Beyond that line, the conditions required to sustain human life exceed what current, or near-future biology, and technology can provide. It is a boundary defined not by imagination, but by the physics of radiation, the mechanics of bone, the chemistry of DNA, and the limits of human resilience. If you appreciate this kind of clear, evidence-driven explanation, subscribing and liking the video helps more of this content reach the right audience. You'll also find many other videos on the channel that follow the same approach.